This video is for The Living Earth, Unit 2.3, Cellular Respiration. Today's essential question: what are the steps involved in cellular respiration? Our first main idea: cellular respiration.
We need energy to power our homes, businesses, and communities. The energy comes from electricity generated by power plants, solar panels, and wind turbines. All living things need energy too. Where does the energy to power our bodies come from? What is our body's power plant? The answer is cellular respiration. Cellular respiration is the metabolic process that breaks down organic molecules to release energy for use by cells. Just about everything a living organism does requires energy. When we think of energy for our bodies, we think of food. Cells use energy in the form of adenosine triphosphate, or ATP, and a reduced form of nicotinamide adenine dinucleotide, or NADH. Cellular respiration is the process of taking the food we eat and converting it into an energy that can be used by cells as ATP. Eating food provides fuel for your body. How? After food is broken down through the process of digestion, food molecules reach the body's cells through the circulatory system. The food molecules fuel cellular respiration. The process of cellular respiration turns glucose from food into carbon dioxide, water, and the high-energy compounds ATP and NADH. These molecules provide the energy for the cell to function. Where does cellular respiration take place? It takes place in the mitochondria of eukaryotic organisms. The process of cellular respiration can be compared to the operation of a gasoline engine. In the engine, gasoline is hydrocarbon. Using air, specifically O2, the gasoline is oxidized and turns into water and carbon dioxide. In the body, you metabolize sugars. Sugars are oxidized by O2 and are turned into carbon dioxide and water as well. However, a cell is much more efficient at using food than the engine of a typical gas engine is at burning gasoline.
Our next main idea: reactants and products. Animals, of which you are one, and numerous microorganisms get their nutrition from organic matter that they metabolize in the presence of oxygen, which is why you breathe, to obtain energy and building blocks to grow, reproduce, and survive. These animals and microorganisms rely on a type of metabolism called aerobic cellular respiration. For organisms that live by aerobic cellular respiration, of which you are one, their primary source of food originates with carbohydrate-rich plants, or other organisms that eat those carbohydrate-rich plants, whose mass and stored chemical energy was made during photosynthesis. So no matter what you eat, the glucose and the stored energy within that glucose originated during photosynthesis. One of the simplest and most common sugars used for energy is glucose, which is again, direct product of photosynthesis. Organisms that get their nutrition from plants then use cellular respiration to metabolize or break down a glucose molecule into then smaller molecules. Energy is released during this process and is used to make ATP. This process takes place through a series of chemical reactions, not just one reaction. In the case of cellular respiration, the primary reactants are glucose, or sometimes a fatty acid or an amino acid, but usually it's glucose and oxygen. Both glucose and oxygen are products of photosynthesis. When glucose is respired in the presence of oxygen, energy is released, and carbon dioxide and water are produced. Therefore, The basic chemical reaction for cellular respiration can be represented like you see on the screen here. One glucose, which is C6H12O6, plus 6 oxygens, which is O2, react to form 6 carbon dioxides, which is CO2, plus 6 waters, which is H2O, and energy in the form of ATP.
Redox reactions. Objective. In this topic, we will learn what happens in redox reactions and classify them. Did you notice the green patina formed on the Statue of Liberty? It was caused by redox reactions. The same phenomenon can be observed in rusting of nails or a cut apple turning brown, and burning magnesium, which gives off a bright white flame. These are examples of redox reactions. In order to study redox reactions, we need to understand the difference between oxidation and reduction. Oxidation occurs when an atom loses electrons. For example, a magnesium atom can lose 2 electrons to form a magnesium ion. Reduction occurs when an atom gains electrons. For example, an oxygen molecule can gain 4 electrons to form 2 oxide ions. In a redox reaction, oxidation and reduction occur simultaneously. When magnesium burns, 2 moles of magnesium atoms transfer electrons to 1 mole of oxygen molecules to form 2 moles of magnesium oxide. Consider the reaction between a strip of aluminum foil and a solution of copper chloride. As the reaction proceeds, the aluminum strip will disintegrate and metallic copper will deposit on the strip. The aluminum atoms each lose 3 electrons as they are oxidized. 3 copper 2+ ions accept 2 electrons each and get reduced to form copper metal. Because oxidation and reduction occur in the same container, this is an example of a direct redox reaction. During the reaction, the temperature rises as chemical energy is converted and released as heat energy. Redox reactions can also be set up so the oxidation and reduction processes occur in different containers. These are called indirect redox reactions. A galvanic cell is an example. The 2 solutions are connected by a salt bridge, which keeps the solutions separate but allows ions to move between the solutions. As the aluminum is oxidized and loses electrons, copper ions are reduced by gaining electrons, which travel through the external wire. Since the electrons must travel through the wire for the redox reaction to occur, chemical energy is converted into electrical energy. Summary: redox reactions involve both oxidation and reduction. Oxidation occurs when electrons are lost. Reduction occurs when electrons are gained. Direct redox reactions occur in a single container. Indirect redox reactions can be set up to convert chemical energy into electrical energy.
Our next main idea, glycolysis. Now, I'm going to play you a video here. Please keep in mind, you do not need to understand this content to this detail, but please follow along the general idea.
Glycolysis. Objective: to describe the process of glycolysis and name its key inputs and outputs. Cellular respiration involves the oxidation of glucose to yield energy in the form of ATP. The first stage of respiration is a process known as glycolysis. During glycolysis, a 6-carbon molecule of sugar in the form of glucose
will be broken down into 2 3-carbon molecules of sugar in the form of pyruvate. The process requires no oxygen and occurs in the cytoplasm of a cell rather than in the mitochondria. Glycolysis begins investing some energy in the form of ATP. A phosphate is removed from the ATP and is attached to the glucose molecule, making it more chemically reactive. This first step of glycolysis results in an ADP and a glucose 6-phosphate molecule. The enzyme phosphoglucose isomerase will convert glucose 6-phosphate into its isomer fructose 6-phosphate. Another enzyme, phosphofructokinase, will transfer another phosphate group from ATP to fructose 6-phosphate on the opposite side in order to form fructose 1,6-diphosphate and ADP. An aldolase enzyme will cleave fructose 1,6-diphosphate into 2 sugar isomers,
dihydroxyacetone phosphate,
and glyceraldehyde 3-phosphate, or G3P. However, another enzyme, triose phosphate isomerase, converts
dihydroxyacetone phosphate,
or DHAP, to glyceraldehyde 3-phosphate, or G3P. Because there are 2 glyceraldehyde 3-phosphate molecules formed at this stage, the following steps will happen twice. We'll trace only a single instance. The triose phosphate dehydrogenase enzyme will shift a hydrogen from glyceraldehyde 3-phosphate
to nicotinamide
adenine dinucleotide, NAD+, oxidizing the G3P and reducing NAD+ to NADH. The same enzyme will add a phosphate to the oxidized glyceraldehyde phosphate, forming 1,3-biphosphoglycerate. The enzyme phosphoglycerokinase will then shift a phosphate from 1,3-biphosphoglycerate to ADP, yielding ATP, and 3-phosphoglycerate. Another enzyme, phosphoglyceromutase, will transfer 1 phosphate from the 3rd to the 2nd carbon of 3-phosphoglycerate, forming 2-phosphoglycerate. In the next step, the enzyme enolase will remove a water molecule, which causes a double bond to form, resulting in a phosphoenolpyruvic acid molecule, or PEP. Pyruvate kinase will then transfer 1 phosphate from phosphoenolpyruvic acid to ADP, producing pyruvic acid and ATP. Overall, during glycolysis, 2 molecules of pyruvic acid, 2 ATP molecules, 2 ADP molecules, 2 NADH molecules, and 2 water molecules will be produced
from 1 glucose molecule.
Summary: glycolysis is the first step in cellular respiration. It is the process of oxidizing glucose in order to form pyruvate and other molecules. During glycolysis, 1 molecule of glucose will produce 2 molecules of pyruvic acid, 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of water.
Our next main idea, the Krebs cycle. Following glycolysis, which we just learned about, the pyruvate or pyruvic acid molecules are transported across each of the 2 lipid bilayers of the mitochondria. Because remember, glycolysis occurs in the cytoplasm, but following that, cellular respiration occurs in the mitochondria. So the pyruvate enters the inner mitochondrial matrix, which is a membrane. Each molecule is then converted to another molecule called acetyl coenzyme A, or acetyl-CoA. This is the starting molecule for the next stage of cellular respiration. In this first step of the Krebs cycle, acetyl-CoA donates its 2-carbon acetyl group, right? It's a group that has 2 carbons, right, in the acetyl. The resulting molecule is called citrate. Citrate is then modified in each of the next steps of the Krebs cycle. By the end of the Krebs cycle, the original molecule that reacted with the acetyl coenzyme A is actually regenerated. And this allows the cycle to begin again and continue with each new molecule of acetyl coenzyme A. There is a total of 8 reactions in the Krebs cycle. Like a glycolysis, each reaction requires a unique enzyme, which again we saw in the previous video. 3 molecules of NADH, 1 molecule of ATP, and 1 molecule of FADH2 are all produced. FADH2 is another high energy molecule and stands for flavin adenine dinucleotide. Again, the H2 indicates the reduced version of the NAD molecule. Remember that there are 2 pyruvate and thus 2 acetyl coenzyme A molecules resulting from a single glucose molecule. This means that for each glucose molecule, 2 full turns of the Krebs cycle occur. Therefore, the energy-containing products from the Krebs cycle are 6 NADH, 2 ATP, and 2 FADH2. Our last main idea, the electron transport chain. By the end of glycolysis in the Krebs cycle, a molecule of glucose has produced a net of only 4 ATP molecules. It is during the next and final stage of cellular respiration that most ATP production occurs. During the electron transport chain, the other high-energy molecules, NADH and FADH2, go to work. The electron transport chain is a series of protein complexes. It is embedded in the inner mitochondrial membrane, and these complexes are electron carriers. NADH and FADH2 donate electrons to the first complex in the series. The electrons are passed down the chain from one protein complex to the next. Each subsequent protein complex donates the electrons to its neighbor one by one, and energy is released at each step. These transmembrane protein complexes also include ion pumps. The energy that is released in each electron passing step is used to pump hydrogen ions, H+, across the membrane. The H+ ions are released from NADH and FADH2 at the same time that they donate their electrons. As these free H+ ions accumulate in the mitochondrial matrix, the ion pumps in the chain pump them from the matrix into the inner membrane space. The 2 final steps occur in the electron transport chain. One might wonder what happens to the donated electrons once they reach the final protein complex of the chain. This is when the critical oxygen molecule becomes a factor. Again, the reason you breathe. Oxygen serves as the final electron acceptor. When an O2 molecule accepts electrons, it becomes negatively charged. It then picks up free-floating H+ ions, forming 2 molecules of water. And then finally, the main question remains, how is ATP produced by the electron transport chain? The answer involves another very special transmembrane H+ protein channel. In the final step, the large numbers of H+ ions that have been pumped into the inner membrane space now form a concentration gradient between the inner membrane space and the matrix itself. This concentration gradient causes them to flow back into the matrix through a channel called ATP synthase. The H plus ions passing through this channel cause a rotor within the channel to spin, and this spinning rotor creates the energy needed to synthesize large amounts of ATP from ADP. This spinning force is only possible due to the large H plus concentration gradient between the inner membrane space and the mitochondrial matrix. So, approximately 32 to 34 ATP molecules are produced during the electron transport chain, and a net total of 36 to 38 molecules of ATP can be produced from a single molecule of glucose. However, in reality, because of various losses in the process, usually around 30 ATP are made available. At this point, you should be able to generate a full and appropriate response to today's essential question: what are the steps involved in the cellular respiration process?